Hello and welcome to this video on buffers. These are what stop your body from becoming acidic when you eat, breathe or drink. They also have an important role in just about any other biological process. For the purposes of this video, buffers are particularly important when we're talking about fermentation. Most biological processes are a very specific example of a chemical reaction. They are controlled and targeted with a purpose. As it occurs in a living cell or a controlled environment, a preferred outcome can be generated by favoring the conditions and resulting products. Not all reactions are so easy to shift the balance for. In other examples, you want to keep the whole reaction at a particular rate, pH or similar for as long as possible. This is where the role of buffers is. They perform an action that keeps the entire reaction or environment at a particular pH. By definition, a buffer is a solution containing a weak acid and its salt, or a weak base and its salt. By having one or the other, it's able to resist changes in pH. By itself, the meaning of this may not be readily understood. The buffer can basically be described as pushing in the opposite direction to a change in pH, but it can only do that for so much. The buffer works by becoming an acid or a base and the salt derived from this. The salt in this situation is the result of reacting the acid and base. By reacting the two and neutralizing the equation, you stop any shift in pH, and this is why they work the way they do. Salts are produced from this reaction, and you have a neutralization of the two components. The importance of buffers in reactions can be demonstrated by two different examples that occur regularly. The two common examples of buffers include tris and the bicarbonate buffer system. The tris buffer is not this, but this, tris-hydroxymethylaminoethane. It is just as reactive, but more easily contained. It contains an amine, and because of this it can undergo a reaction, and this is true of just about all amines, that produces a salt and a consequential byproduct, whether that is the acid or the base depending on which way you're trying to push the reaction. The bicarbonate buffer system is basically the consequence of you breathing. You produce CO2 because your body makes CO2 from every process that it goes through to keep you alive. CO2 then gets dissolved in your blood. The blood then turns it into one of two forms. It's either going to be bicarbonate or carbonic acid. In these two forms, you then have it further reacting. It's either going to gain or lose a hydrogen. The gain or loss of this hydrogen is what allows the acid to turn into a base. That is your neutralization reaction. It allows your body to keep your blood pH between 7.35 and 7.45. These two examples show how a buffer can stop a reaction from running out of control. In the case of tris, it's useful for keeping a neutral environment for blood, medicine, cells, and other biological processes. The bicarbonate process is necessary as it keeps your body from turning into a molten mess, particularly as acidification would melt you from the inside out, and that's generally not advantageous to life. In terms of other benefits it brings, it ensures that your body remains at a stable level as far as pH is concerned, not only around 7.4, but as low as 7.35 and as high as 7.45. These lessons can then be extrapolated out to other processes, particularly brewing. Notably in brewing, beer is an area where a buffer can be very useful. The addition of acids and ions can help to minimize changes seen in your fermentation as it proceeds through that process. Beer and wort is the most common example, as the fermentation of wort into beer needs a certain amount of buffer added to it to mitigate the various reactions that you see in solution. Common examples will be something like the bicarbonate buffer solution or system. This involves the use of carbonic acid and bicarbonate. Things like phosphoric acid and phosphates also work in the same role. All of these can create pairs of a weak acid and a weak base. When you put them together, they then work basically with each other and against each other. The advantages of this is that you can keep your wort and then later fermentation at a much lower pH that would eventually be neutralized out. This means something like amylases that work really well at a 5 pH can be kept there for much longer. or when used judiciously and correctly, they can have great effects on changing the ability to use the material while not altering the flavor and outcomes in your fermentation. This is why they are very useful. In summary, a buffer is generally a liquid and it keeps the pH of a solution roughly the same for most of the time it's going to be active. The buffer is made up of two parts, a weak acid and a base, or a weak base and an acid. These two work together. When you react the two, you will make a salt. And second to this is that these can be added before any changes occur in the solution. This means you don't need to be reactive but proactive. With that in mind, you can add things like a buffer to your wort or to your extraction and get a lot of benefits in advance without having any negative effect, and therefore make your life that much easier. The nature of many fermentations, for example wort in beer, is already going to be taking advantage of this to some effect because there are amino acids in solution already, and these will undergo the reaction that we're talking about and produce their own buffer to some extent. The downside is that they have a limited range at which they are effective. Once the pH is pushed beyond that point, the buffer no longer has any capacity and will stop working, and this is perhaps the only notable downside to them in most cases. Once you exceed their capacity, they no longer have any role to play. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below,